Falling Walls Circle, Challenges and Foresight. Welcome to the Falling Walls Plenary Table. Altering Medicine by Cell and Gene Therapy.
Kann ich? Guten Morgen. Guten Morgen, mein Name ist Johannes Fruehauf. Welcome to our plenary table on cell therapies and gene therapies and innovation. Cell therapies carry a lot of promise for patients worldwide to treat devastating conditions. They benefit from close collaboration between academic inventors, pharmaceutical companies that carry them forward, and regulatory agencies. These are completely novel modalities for which there isn't really a precedent. So we need effective collaboration between these different players in the innovation game. In our plenary table, we have academics, we have industry partners, and experts in delivery of cell therapies. I look forward to an open discussion with all of you and look forward to engaging the audience as well to see what we can do to break the walls of medical innovation for cell and gene therapies. Thank you. My panel, please join me. I'm thrilled to welcome at this table Professor Hildegard Büning, Professor in Infection Biology and Deputy Director of the Institute of Experimental Hematology at Hannover Medical School, Debora Lucarelli, CEO at Enhanced Genomics, Professor Wolfram-Hubertus Zimmermann at the University Hospital in Göttingen and head of the Department of Pharmacology and Toxicology in Göttingen, Sarah Hedtrich, professor at BIH here in Berlin and with a focus on gene therapies. And last not least, Dr. Maria Alfaiate, Senior Vice President for New Products, Commercialization and Portfolio Strategy at Bayer Pharmaceuticals here in Berlin. Welcome and thank you for joining us this morning. Maybe to get us started, the objective for our panel is to really discuss how we can, in the field of cell and gene therapies, enhance translation from academic findings into products that will arrive at to benefit patients. Hildegard, maybe we'll start with you. Can you give us a quick background on your own work and how you see this unfold?
Yes, thanks. So it's really a pleasure to be with you and to discuss on this very important topic. So in my science and research world, I'm dealing with developing viral vectors for cell and gene therapy, in particular focusing on getting them in vivo working. But I think we have to do much more than that, and this is also to be engaged on different levels to move this very, very important innovative therapy forward to the patients.
Debora, you're the founder and CEO of Enhanced Genomics. I read about your work. Can you tell us how the analysis of the genomic structure will help develop individualized therapies for patients?
So today a lot of therapies are really inefficient because what we are not taking yet into account is the precision medicine aspect of it. We are all genetically different and therefore we need to take into account these factors when we think of a treatment. And in the field of gene and cell therapies, this is obviously a very important factor. So by utilizing genomics information that come from patients in particular, we're trying to further the field in this direction and design some panels and some therapies that are a little bit more personalized for the specific needs of a disease. As a very simple examples, each disease contains many subgroups of many diseases because we don't all react or respond in the same way. So we tend to start from these points and then we start with gene modulation approaches in order to facilitate the development of treatments.
Thank you. Professor Zimmermann, you are a pioneer in tissue engineering and tissue repair, and you run a very exciting trial to help treat patients with myocardial infarction. Can you give us a background on your field and what you bring your view of from the academic world and translation into products that arrive in patients' hands?
So I've been in academia since 1995. Started as a medical student, actually, working on a topic that was at that time not existing. That was the development of tissue, which may eventually be used as a screening platform for compounds, but also as a tissue which could be implanted to repair the heart. And this is what we have been doing now since 25 years. And of course, there have been many challenges at the interface of academic development and moving academic developments towards approval by regulatory authorities. And eventually, of course, also we have to deal a lot with questions how such a product in the end can enter a market. And that's basically what we are working on.
I can't wait to hear when the first patients will be treated with this. Professor Hedtrich, you are an experienced academic scientist working at the cutting edge of gene therapies and payload delivery. I know your work also involves the delivery into targeted tissues. There are many applications for this. Can you give us a quick overview of your work and how that will lead to translation and arrive in patients' hands?
Yeah, of course. So my lab is really focused on delivering new gene therapies to human epithelia and specifically into the skin and into the lungs. And this has not received much attention so far. So mainly, most of the gene therapies target the liver so far because it's also easy because everything ends up in the liver eventually. And so we are really trying to utilize and develop also smart tools, carrier systems that help us to get it into the tissues of choice. And this is a very exciting field. And what we do in order to better bridge bench and bedside is that already in our preclinical work, we are almost exclusively working with bioengineered human tissue models, which really resemble human tissue instead of classic animal models, which very often fail in recapitulating human diseases and human anatomy and physiology.
And so we'll hear about how especially in the field of cell and gene therapy, new models will be useful and needed to predict efficacy. Dr. Alfaiate, you're a pharma executive with many years of experience working for Bayer and bringing products into the clinic and then commercializing them. I think this is a very important viewpoint. Can you tell us a little bit about your company's activities in cell and gene therapies? Bayer is known as a small molecule company. They sort of missed the biologics revolution and now they're doubling down on cell therapies. And there are exciting initiatives inside of Bayer that hopefully will make sure that these products arrive in patients' hands.
Absolutely. Bayer's vision is health for all. And of course, if we had remained as a player only in small molecules, there would be a lot of the population for whom we would not be able to provide health solutions. So it is true that Bayer took the challenge of becoming a company that also now works in these specific technologies. We have acquired recently 2 innovation engines in cell and gene, and we're working very closely. We're observing the space, and we're seeing how in the future these drugs will have to be developed in a slightly different way from what is the legacy Bayer way, so to say. So on the one hand, we are trying to keep up with the exciting changes in the environment, and some of them by the distinguished members of the panel, and at the same time trying to bring the expertise of what does it take from a regulatory point of view, from an access point of view, what do we need to take into consideration to make sure that one day the beautiful science is able to treat patients.
Can you talk to us about what you have in plan for Berlin with your translational center?
I can indeed. So Bayer is working with peers such as the Charité, BIH, and the city of Berlin to establish a cell and gene translation center. So we're quite excited about it, quite excited about the possibility of making Berlin such an important hub, not only for Germany, but for Europe. That is the level of ambition. Things are ongoing, so we expect to have good news soon.
Yeah. Soon being a relative term.
Soon is a relative term, yes. I know everyone probably wanted it to be yesterday. It's not going to be tomorrow, but we're confident that this is the right thing to do. We have the contracts ready to be signed. We understand exactly what funding is needed. So things are in place, and now we just need to get going.
So the center is supposed to help with the close integration. It's also in collaboration with Charité and BIH to facilitate really the smooth translation of new academic findings into products. Here, I'm very excited to have Professor Zimmermann with us, who is an academic who is doing this himself now. Can you talk to us a little bit about your ongoing trial to use iPSC technology to ameliorate cardiac conditions?
Yeah, of course. So we have started in 2014, actually, to engage very carefully with regulatory authorities to discuss the steps required to bring our approach as a product into a clinical application. And that culminated in the approval of our clinical trial in 2020, December 2020, and the first treatment of a patient in 21. So we now have close to 3 years follow-up in total 11 treated patients, roughly 7 billion iPS-derived cells implanted. We have done that through our academic institution together with a startup that we have formed called Reparon, and very closely also with the University Medical Center, not only in Göttingen, but also in Lübeck, to be able to address the patient population that we're presently targeting, and that is patients with advanced heart failure, which have no other treatment option, very often candidates for heart transplantation or for mechanical pump implantation. And this is the patient population we are presently treating, and the first data from the clinical trial will be presented on Sunday in Philadelphia.
So many of our audience members may know cardiac failure as a condition with their elderly parents or relatives. So am I imagining this as a treatment that will be available to all patients with heart failure, or is this for a subpopulation only?
So in heart failure, the fundamental issue in heart failure is the loss of heart muscle cells. And it's very simple. It's a very simple approach or very simple thought to think that if you lack muscle cells, you have to put muscle cells back into the heart. This sounds very simple, and actually it's not so simply done, but this is the concept we have been working on. We have been trying to solve all the issues that are in the way of achieving this relatively simple approach. And in the end, the concept is, and our hypothesis is, that the more cardiomyocytes, heart muscle cells, that we are able to bring into a failing heart with the correct orientation and function, the better the heart function will be, and the better the pumping action of the heart will be, and ideally also the symptoms in those patients will be reduced.
In many cases, these innovations, for example, the new cells, are developed in academia, in academic centers, often in collaboration, but they do need to eventually be moved out of academia into commercial entities. And you mentioned that you started a company on the side to help with that as well. Debora, you started your own company. As we are trying to think about how to improve translation, especially in Germany, of what we know is excellent academic science, but a lack of effective translation, what are some of the learnings that you can share with the audience from your own path, from being an academic to being a founder to being a CEO?
Yeah, and it's possibly one of the reasons I have left academia. I mean, I've obviously, like most people, I've started in a high-profile institute doing PhDs and postdocs, and I got very frustrated with the translation speed of your research. Papers are interesting, but maybe they don't go too far. There is a lot of things to take into account. When you think of how am I going to translate an idea and who is going to be the recipient of this idea, I've learned that the most important thing is to think about the market and the final product that we're trying to develop and who is that going to be beneficial for. One thing that is very obvious is that in order to translate science, you need to have enough evidence to bring it to the market. Collaboration is one of the most important things amongst academia, but as well industrial partners. So this is the first steps. You need to gather enough information, enough evidence to create the proof of concept that can convince people that this is going to be a viable product. And then there are many stakeholders that we need to engage along this journey, not just academics, but then we start to engage people, venture capitalists or funding bodies that they are prepared to come along with you, doctors who of course will have to be the ultimate drivers, you know, from the interactions with the patients, and the regulatory aspect, because every time you have an idea, people always want to work, how are you going to bring it to the market? For this, we realized that there is a lot of partners to engage. There are pharma partners, but there are likewise the NHS in the UK, for instance, has actually a lot of interest in facilitating this kind of innovations. And therefore, there is the possibility for innovators to work together with collaborators and create studies or piggyback on other studies so that some of these ideas can be brought to market a little bit sooner and have a real effect on patients.
Yeah. We hope to learn, in Germany, we hope to learn from the UK and from the US. There is an aversion in many academic centers to allow for dual activity. Yet I personally think it's so important that the champion, the inventor of this technology, be the one that takes it forward, because they're most passionate about it. And especially if we're talking about a field like cell and gene therapy, where we have so many novel developments, and there's still expertise that needs to be built inside of the pharma industry. It's not like a small molecule development where you know what to do with the molecule and you toss it over the wall over to pharma. Here we have a field where the academic inventors really have the ability, should have the ability to carry this forward. Sarah, I know you work on genetic payloads, you work on particles. What do you think will be the first applications for gene therapies going forward? What are you most excited about for the next 5 or 10 years in your work or in the work of your peers?
Yeah, I think we are already seeing huge advancements right now. I mean, there are already clinical trials going on for genome editing for certain types of blood diseases. And you know, if I remember correctly, the documents have already been submitted and they are now currently being reviewed by the FDA. So I'm sure that we will see very, very soon, likely sometime 2024, the approval of the first gene-edited drug or therapy, which is, you know, super exciting. And I think, you know, we are currently seeing an unprecedented growth in this area. I mean, 2018, there was the first approval of an siRNA-based drug. Then 2020, another landmark decision where the COVID-19 mRNA vaccines. And this together also with the, the Nobel Prize 2023 in the area of mRNA modifications, this all together, there's a huge excitement about that, and many people are, you know, joining forces and pushing for advances in the field. And I think we really have the unique opportunity to provide patients that we couldn't treat well in the past, for especially people with rare diseases. To provide them for the first time with an effective therapy and potentially even a cure. And this is super exciting. And I also think, and I hope, but again, we need to join forces, that we can move fast. That, you know, but my understanding is that regulatory authorities, pharma, is on board with that. But there's many question marks remain, so many unknown things, right? Also when it comes to safety. And so I'm very curious how this will pan out.
Yeah. In drug development, you want to be the second. You don't really want to be the first to educate the regulatory agencies. Hildegard, I know you're a member of many professional organizations in the American Society for Cell and Gene Therapy. So you see a lot of things. We heard from Sarah. What are your what are the developments that you observe? I know you're working on targeted deliveries as well. Where do you see the field moving in 5 and 10 years? What are you most excited about?
Yeah, so let me begin like really saying the same words as we heard a minute ago. We really have to look where we are already. We have gene and cell therapies on the market, which means available for the patients with regard to monogenetic diseases, with regard to cancer. And when we now come back, gene therapy was initially really thought of to be an innovative therapy developed for monogenetic diseases, but we go far beyond that. Infectious diseases, cancer, as I mentioned, are only some examples, and we go more broad and broad. This is, I think, already an achievement we have. And now we have to look into what we have and really to see how we can make them accessible and maintaining them accessible to the patient once they are really on the market. And then we have to go back to research and see how we can improve. We heard about safety, but we can also think about moving from maybe ex vivo into in vivo by optimizing the delivery tools in a way that they are passed really only to those cells where really the therapy has to be applied. Yeah? Be it gene addition, so you add a novel copy of a gene, or being genome editing, as we heard a minute ago, for really precisely correcting the defect. So I believe we have to look into all these, making and seeing that it stays accessible and bringing novel paths with regard to really opening the field even broader as we have it now.
I want to double click on the topic of accessibility, because that's sort of the elephant in the room. Yes. Accessibility is code for affordability.
Yes.
Who can afford these therapies? The Kymriah first approved CAR T cell therapy from Novartis, which was spun out of UPenn. It costs Novartis to make more than $500,000 per dose. It costs them like $600,000-700,000, and these are the pure costs to produce. It's impossible to imagine that we as a society can afford these types of treatments when they clock in at $1 million per patient or per treatment. This is an open question to all of you. How can these problems be solved? What are the approaches if the therapies are wonderful and promising, and they may lead to a cure, but we can't afford them?
I can start, just because I have the solution. No, I think it will take everyone, right? We will need everyone's help. On the one hand, there's going to be an element of scalability. We are currently dipping our toes in the water. These are usually smaller populations. So of course, there's a lot of trial and error still involved. So of course, the costs of producing these drugs, these interventions are very high. As we move forward, as we are able to de-risk the technical components, it will also be easier to find scalability options, and therefore producing them will become cheaper. That's the one side of the equation, and that's everyone's responsibility. The other side of the equation is how do we prepare healthcare systems to actually cope with thinking about something that could be a one and done. So we're not talking about a patient who will be receiving, let's go with heart failure, right? A patient with heart failure, from the point of diagnosis, 50% of these patients will be dead within 5 years, despite great therapeutic options on the market. But what we're talking about is not having the patient for 5 years on a number of different small molecules, still with very poor results, moving them onto a device, still with fairly poor results, not only in terms of keeping the patient alive, but of their own quality of life. We're talking about an intervention, hopefully, that is one and done. So the system is still not prepared to assess fully the benefit that this will bring. So I think that it's a two-pronged approach. On the one hand, scalability will make the interventions cheaper, but the systems need to think about how that is an alternative to a different way of using healthcare resources.
Very, very important. You know, we have presently therapeutics which are very cheap on the one hand, and on the other hand, we also have very standard small molecules which run at costs of roughly 300,000 euros per year. And if you apply that for 10 years, this will be largely more expensive than what we are talking about today. And the small molecule approach is much more acceptable for now. But I think it's exactly as you said, once we are able to demonstrate that a therapeutic would then cure or repair a certain disease. So this should be a paradigm shift, actually. We are not aiming at, we don't want to delay disease progression anymore. We want to cure. Yeah? That's a huge goal, but we have at hand methods, be it gene therapeutics, nucleotides, cells, to repair. Repair is the first step to cure. This is how I would see it. And then it will also be affordable. Of course, the production has to be optimized. This is one thing that everybody is working on, to scale production. I'm pretty confident that the prices will fall.
Maybe something that I can add as well is talking about the delivery tools. So we heard about viral vectors, and the very first gene therapy, Debora, was viral vector based. Effectively, it was taken from the market, not because it was not effective, but upscale and production was super expensive, and exactly what you're saying, it was not affordable. However, just alternative non-viral delivery systems, we see a huge increase in that, talking about lipid nanoparticles, for example, that are very efficient in delivering genetic cargo, and they can be easily produced at a fraction of viral vectors. And so I really think by investing in that and making those delivery tools better, we can also cut costs significantly.
I think we are hearing as well, to add to this, that there are a lot of options here, but none of them seems to have gone particularly far, or there is not enough data to support all of them. And I think in time we need to be able to stratify the type of cure that we can offer to certain people. And going back to what Maria said before, we need to have tools and enough data and information, and one person can do it all, not even a massive pharma, to generate that resource from which we can learn first and understand how this method works, and then from that we can diversify the various approaches in terms of therapeutic treatment or selection of the patients, because otherwise everybody seems to fence a little bit on his own with his own tools. And then there is also the other side. We have a lot of conversation with clinicians, and then coming back to what you just said, they need to almost understand and retrain a little bit on the therapeutic opportunities that they are available, because sometimes it's not easier also from a clinical perspective to advise a patient to go for this treatment versus this other treatment. At least in the UK, we see this is not just about being available as one of the therapeutic options that they can offer, but it's actually understanding the very complexity prognosis that is behind, and then put the patient in the right path.
And I mean, this is particularly important. So on one hand, of course, the physicians who will apply the therapy, that they really understand what they are doing, so really in deep, and can really also advise the patient. And then also on the other hand, we have to look into the patient organization groups and really to explain everybody exactly what the therapy is doing. Yeah, where are the risks, where are the benefits, that really also will help to really understand where are still the barriers, but what also these innovative therapies can do, and where the future is. And for example, also if we think about gene therapies for monogenetic diseases, we have to be aware that also these patient population in most of the cases do not have the time. You have to be really very quick with applying this therapy. Yeah, so it's really clear that we have to make the teaching of everybody and the training of everybody really ahead. Yeah, that's very important also with regard to accessibility and the mind setting.
We've had cases in which questionable gene therapies have been pushed by the parents' associations, who are, of course, desperate for their kids. In DMD is one example, right? And then there are other cases where drugs that were approved in other jurisdictions and approved in Europe are now no longer paid for in Germany, I think specifically, because of the outrageous cost. I call $1.5 million outrageous per patient, right? I know, Maria, you've had some discussion about that. Can you help us work through this thicket? You're working on the commercial side, on the regulatory side for a big company.
Yeah, it's, I mean, different, the same way different regulators look at different things. So the FDA has an approach that is not exactly the same of the European Medicines Agency. The way that drugs are reimbursed in the US and in Europe is very different. In Europe, most of the countries has a single payer who has full power, whereas in the US, different payers may decide to reimburse under different circumstances. Versus the, I'll call it totalitarian approach, which is what I say goes. But at the same time, I don't want to be unfair. At the end of the day, the mission of these systems is to provide solutions for as many patients as possible. So it's the old blanket analogy. If you cover your neck, your feet are out, and if you cover your feet, your neck is out. So of course, they're trying to distribute the resources available in the best possible way. The process is relatively predictable, which also puts some of the strain on the drug developer. So if I know that this is the type of evidence I need to put forward to justify the value, then it's my responsibility to produce a comprehensive data package. And in some cases, that is not there. There's also the need for policy to change slightly. There's the need for some of these reimbursement systems to accept risk-sharing agreements, which are more common in the UK and in the US, whereby there is reimbursement of a particular drug, and then it is reassessed. And if it's not working according to the expectation, then the drug developer needs to pay back to the system. So I would like to think that we will get there. We will get there in finding ways to make the drugs available to as many patients as possible at a price that is responsible and appropriate to cover the development that we've heard is not easy, but at the same time that the system recognizes that with many of these interventions, we're not trying to build something on top of each other, we're trying to displace years of little chipping at the risk without actually being able to modify it with something that is groundbreaking.
Yeah, I think societies have to come to grips with this novel world, right? It's no longer you go to the doctor and you get a little pill and it fixes you for the next week or two. We talk about the C word in cancer that you shouldn't ever mention in front of the patient because it gets their hope up, right? But if we can cure the disease, that is very different from managing it through side effects and clinical negative outcome of the treatment. If we think about a paradigm where a patient with end-stage heart failure may no longer have to wait on the list for a transplant organ for many years, and instead with a cell therapy intervention might be cured or to a place where their quality of life is much better, then we need to think about the appropriate ways to reimburse for that. I know that there have been models for gene therapies that would be sort of pay as you go, where instead of charging the system $1 million or $2 million upfront, you would sort of pay per year of proven benefit to the patient. But these are always fraud ethical considerations, right? Who owns that patient? Who owns that patient's health if you have to sort of make an ongoing payment to maintain that?
It's one of the difficulties in the US because patients may move from one insurance provider to another one. So those types of risk-sharing agreements, although they're exciting, they pose the issue of how do we track it? Who has access to the data of the patient? How do you deal with data privacy? So these new treatments are making us ask ourselves questions that we hadn't had to think about before.
So I was asked to leave time for audience questions. We didn't even touch upon my favorite topic, which is how to make it easier for academics to really translate their questions. Adam, I know you want to take over with a couple of questions. I'll push it over to you.
So just a reminder to our online audience, drop your questions in the chat, and to our audience here, raise your hand and we'll make sure to get you a microphone if you have a question. But to get us started, how do public perceptions of these kinds of therapies influence how we can actually roll them out? Do they present any barriers to us? Are there fears around these kinds of therapies that we don't face for other kinds of interventions?
I think that in particular the professional societies, like here in Germany, the German Society of Gene Therapy, or for Europe, the European Society of Gene and Cell Therapy, are in particular keen really to give an unbiased opinion on the cell and gene therapy. Where are we? What are the risk-benefit, as I was speaking a minute ago? And this is really our responsibility as researchers, really to show where we are. And we are still doing science. We are still doing research because we would like to improve the system. But at the time we are here, we already can really provide therapies. And so I think this is the responsibility of, I think, amongst different other stakeholders, but also amongst the persons involved in these professional societies to really unbiasedly show what is possible and what not.
Maybe I can add to that. I mean, we have seen the controversy around the mRNA vaccines, I think worldwide, right? And this already showcased the controversy in the lay public. So I think it's very important that the proper science communication is done, and we are also lacking that in Germany, in my opinion. And on the other hand, I'm in touch with many patients, monogenic diseases that cannot be treated today. And they are super supportive of that. And I think, when it gets to the point that you don't have any options left, people are they are supporting science, and they are also very good educated, and they are also willing to accept certain risks, because right now we touched on safety, but I think this is a two-sided sword, right? So on the one hand side, we need to do proper science communication. There's always controversy around gene therapy, but on the other hand side, there are those in need that are really behind it and are pushing it, and are also great motivation for us scientists.
Maybe I can add to this. The UK has a slightly different system, obviously, compared to the German one. And there are some groups well advanced these days. There is obviously a lot of patient advocacy groups, and there are a lot of charities, the Alzheimer's Society, as an example, and many others, that they take on board some of the patients' needs, but as well the communication across the general public. As well as there are institutes like the Cancer Research UK that they're really focused not only on the translation, actually, not just the research, but the translation and the actual therapeutic application for some of these approaches, and they take on board to become advocates somehow for the entire country, for the general population. So there are ways actually to involve the public, but it takes time again, and somebody needs to invest in this type of communication strategies, for instance.
It's completely clear. Patients' voices have to be heard. This is one thing. And the other thing is, of course, that regulatory authorities also have to be involved very early, and they have to, and we need to put also some trust in the regulatory authorities, much more than we have done from the academic side in the past, because these are the regulators that approve a product, and they must check whether or not the data is sufficient to ensure safety. And this is what really has to be happening much more, that academia and also the national, international institutions and groups like the European Stem Cell and Gene Therapy Society, they have to work very closely with each other, and also take into consideration that there are differences between different nations. And it's not, EMA doesn't decide, or FDA is a different story, but in Europe at least you have still a big diversity of opinions also. And each of these opinions are heard. And of course, if you want to do a therapy in Germany, then the German regulatory authority, which is the Pauli Institute, and has to ensure that everything you do is according to the German drug law. And that is a big challenge.
But I mean, in particular, cell and gene therapy, because when we again look into monogenic diseases, because of the size of the patient groups, also is a prime example where we see that collaboration between the different nations are in particular important, and this is lived, yeah. Despite exactly what you said, that we have national regulations, but this communication, the exchange, and the willingness to make it possible in different countries is there. No? And this is also something which is unique, I think, in cell and gene therapy.
This is fantastic to see in Europe, that these different regulators are discussing all the time. And of course, they are responsible for an approval in their specific territory. So in the end, in Germany, the PI has to decide, and not the EMA. Yeah? And that's important to realize.
It's interesting how our expert panel pivots to regulatory question when the actual question was the public perception. I think this is fantastic to have a full audience here today to show that the public is actually interested in this. I assume that not most of you are regulatory experts, but you may be interested in this because you're a patient or you know someone who's a patient. I think the public discussion is scary. If I think back at the anti-science discourse that happened in America and also in Germany when the vaccines came around, which is a wonder of science that we were able within a couple of years to have drugs developed that are effective and safe. And yet we are facing a public that's very skeptical of science because they haven't been taken on the journey. The science education hasn't been there. It's not broadly considered cool to be a scientist. That's where it starts.
Sorry, if I may. I think part of it is also what Sarah was mentioning. If the public were to understand that regulators, and going back to our favorite topic, but regulators have patients' interests, societal interests at heart. Every drug has to do more good than bad. It's a simple equation. Of course, they understand there will be, it will not behave in the same way for everyone, but I think that for the public, this should be reassuring.
Let me just jump in because I think we have a question from an audience member over here.
Yes, sorry. I'm a clinician and I'm really struggling with digesting all the rapid developments and changes that are currently happening in this space. And I feel like there is a large onus on clinicians to not only digest the science, but how rapidly it's developing and then communicate that safely to our patients. Is there any world where you imagine cooperating more as pharmaceutical companies to give streamlined advice regularly to clinicians rather than us individually seeking that out?
I think that we more and more realize, we as researchers, that we really have to, that our duty and our heart is there to really speak about what we are developing and really to educate in a way that you then as a physician can really judge on yourself so that we are somehow, and this is what I mean with unbiased training, that we really bring the information accord, but on the other hand also don't leave you alone later on if the drug is on the market. So can I really apply this to this specific patient or not? And I think this has evolved in a way when the first market approval started, because we are also learning on the fly. Yeah? And when I speak to my colleagues, everybody is really keen on helping in this regard and to set up more and more educational activities. And I can only let you know, just encourage us on that way, because people are there doing it, yeah? And they aim to do it.
Absolutely.
I think we have time for one quick question from this audience member over here. Hopefully there's a microphone we can get to this person. Thank you.
Thank you so much for a really, really amazing discussion. So I'm wondering a bit more about like perhaps it's coming from my own ignorance, but so there's a lot of potential side effects with any therapies, like small molecules. You have unintended side effects with gene therapy. You can have misproduction of the protein targets with cell therapies. Maybe the host is still attacking its own cell. So the research for those kind of therapies are especially tricky. Is there any way that you guys believe that we can improve the personalized medicine to say, okay, our therapy will work for this patient, but not for another patient with the same disease, because you have this expanded knowledge?
Yeah. We are big believers. So we obviously have a company, a commercial company that does that. We believe, and we have actually evidence, that using your genomic information and many other marker, multi-omics targets and expressions, you are able actually to already stratify patients. You will have some sort of indication already from your initial personalized profiles that some therapeutics will be better than others. And this is due to the fact that biology is very complex. It's not just about your DNA. The cells talk to each other. And so hence, you want to have a bit of an idea which cell therapeutics you want to go for. Is it going to be a beta replacement therapeutics, or is it going to be a beta depletion therapeutics, and so on and so forth. So there are many things that people can do up front in order to de-risk actually the pathways. But again, you need a lot of data, and you need a lot of information in order, and you need to make a lot of patients' data again available for people to develop these so-called panels. And you need to work with clinicians like Anna, who just ask, how am I going to choose what kind of therapeutics I'm going to treat? Why is this set of genes or panels or marker or biomarker is going to help me identify that this drug or this therapeutics is going to work better for patient A and patient B. So it's possible to do, and there are lots and lots of evidence and a lot of studies. But again, everything has to come together, not just the therapies.
Maybe to add to that, many of those gene therapies, be it especially genome editing, is very much tailored per se, because it really depends on, even if there is a certain gene that carries a mutation, it's in most cases not one mutation, there are several mutations that cause the failure of the gene. And so, when you're developing the therapies, you automatically make it a personalized therapy in the way that you tailor it to the exact mutation of the patient, and then you automatically know, and you can extrapolate, that this will help this specific patient. So what I'm trying to say is, it's already the gene therapy, all cell therapy engineering approaches are much more tailored and much more personalized than all those biologics and small molecules therapies ever have been.
Sadly, that's all the time we have for questions from our audience, physical and online, so I'll hand back for some concluding thoughts.
Thank you, Adam. I want to say thank you to the organizers for allowing our panel to pull back the rug a little bit and uncover a little bit the excitement that is in our community about these novel modalities. We've heard from experts in clinical medicine, we've heard from experts in delivery, and we've seen that it's this novel, promising field is a complex challenge to all of us. Basic science, as well as commercial and regulatory, need to work together in order to make effective therapies available for patients. I want to say a special thanks to my esteemed panel, and please join me in a round of applause for the panelists. Thank you.